this one in particular is brand new, but we have another that's on a platform like this. And you very much want to spend some time with one of those two models. From left to right, you are looking at the kidney macro view, meaning from far away. Then we get in, and with greater resolution, we look at the structure of one pyramid, and the collecting tubule system, which is represented in white, and the vascularity, or the vessels that serve this portion of the kidney tube, represented in red and blue. Then you move to the far right, and you look at one nephron. The nephron is the functional unit of the kidney, and the purpose of the kidney is to filter the blood. So this is where that's happening. And specifically, I have my hands on the Bowman's capsule portion of the nephron. This is the fibrous portion that provides support for the pressure dynamic that we see in here around this structure called the glomerulus. So far away, closer, very, very close, microanatomy. Macroanatomy, somewhere in the middle, micro. Here's the kidney as a whole. This is very similar, the view, to what we have here, so that you have two things that you can access for anatomy of the kidney, macroanatomy. Okay, let's look at what we've got here. We have one renal artery and vein serving the kidney. We see this large tubular structure moving into the kidney that is right here at the number 13, called the renal pelvis, where it enters into the kidney itself. These, what look like upside down funnels, are the collecting tubules that help to collect the filtrate that's being concentrated in these pyramids and conducted away from the kidney. So we call these calyces as they approach the pelvis. This would be the minor calyx, or the first one that we see at the bottom of one pyramid. Then we'd have the major calyx, where the minors merge. And then we would have the pelvis, where all of the major calyces merge to actually exit the kidney itself. Remember from our discussion with the cat, two regions. There's the outer cortex of the kidney and the inner medulla. The inner medulla is made up of these structures called pyramids. They are roughly conical when seen in 3 dimension. They look pyramidal when you see them in 1 dimension. So here's one that you can see looks very conical. That conical shape, or cone shape, helps to conduct the filtrate that's being formed here into this collection system, and helps it to exit via the ureters to storage in the bladder before it leaves the body. Let's look at what do we find in these pyramids. Usually we'll have roughly 8 pyramids. That number will vary somewhat. And you see them in this model here also. And they almost look like a chrysanthemum in this model, a flower that hasn't opened fully, when you see them partially dissected around the edges. Let's look inside one of these pyramids by moving to the middle portion of this model and see what we have. This is the part that will give people difficulty. That part's pretty easy to understand. Here's one pyramid. Here is the minor calyx, ready to collect all of the filtrate that will be coming out of these pores. The white system that we see here is what we call the collecting tubule system. Let's find one right here, Bowman's capsule, and follow the path of filtrate formation. Let's describe the filtrate formation and then follow the path of that filtrate, which later is called urine. Right here in the middle of that Bowman's capsule, we have the glomerulus, the capillary net, viewed here more expanded or enlarged. And that is where we see filtration happening. So we have this afferent vessel, afferent approaches, coming into the Bowman's capsule, supplying it with blood. Here is that afferent vessel coming in. Note the diameter of that vessel. It is bigger than the exiting or efferent vessel, which is of smaller diameter. That's really important to maintaining the pressure dynamic in the glomerulus and modulating the rate of filtrate formation. So afferent is larger, afferent approach, efferent smaller, exits. Once we create filtrate in this negative space around the glomerulus, it moves into the beginning of the tubule system, and it's very convoluted or wiggly, if you will. And we call this the proximal, therefore, convoluted tubule. Proximal meaning it's close to what? The Bowman's capsule, where we just started. So that's called the PCT for short. And then it straightens out and dives deep down into this pyramid here. And that is the descending portion of the loop of Henle. The loop of Henle is what we call this straight hairpin structure that dives down into the pyramid. What do we see happening here? Without giving you too much physiology, but to help you just understand the structure, we have formed filtrate in the glomerulus. It's been collected by this Bowman's capsule, and it leaves into the PCT, or proximal convoluted tubule. Remember from cell biology, is filtration a selective process? Meaning, can we pick and choose what moves across when we filter? No, things go across that you don't want to have go across, because it's just a pressure-driven process. So that means that there will be solutes and things in here that we don't want to get rid of. So this tubule system's job is to reclaim anything that was filtered out that we would like to keep in the body. Also, sometimes we want to dump excess into the filtrate or the urine. Excess bicarbonate, excess sodium, drug metabolites, these kinds of things. So this is the beginning of the process in the proximal convoluted tubule. Dive down into the loop of Henle, turn the corner. Now we're in the ascending portion of the loop. We come all the way back up to the cortex, and now it curls again. It's convoluted, not as much as the PCT, but still we'll call this the DCT, or distal convoluted tubule. So here, this entire structure represents the nephron. We have one right here, close to the medulla. That's called the juxtamedullary nephron. Juxta means close to. It's close to the medulla. Note that on this rather tree-like structure up here in the cortex, we have other Bowman's capsules associated with nephrons, but they're further up, more superficial. They're further away from the pyramid, the medulla. And so these are not juxtamedullary. These are cortical, because they're way up high in the cortex. So you've got two types of nephrons based on their proximity to the medulla, the juxtamedullary nephron or the cortical nephron. Note the length of the juxtamedullary nephron's loop of Henle, is much greater than the length of the loop if we're talking about a cortical nephron. Alrighty, so when we really refine the solute concentration of the urine, it's because of these juxtamedullary nephrons. And this longer loop represents more opportunity to fine-tune the concentration of that filtrate.
Why is one shorter? Why is the cortical?
For that reason, some are not as concerned with fine-tuning the solute concentration of the urine. So we need to drive and produce and filter, but we don't need to have every single nephron involved in fine-tuning the concentrate. Now, what else do we have here? We've got all these blood vessels draped around these collecting tubules. Now, the older model that we have on Island 6, will somebody go grab that one with the black base for me? That is really the best model for understanding this part of the discussion. If I'm telling you that we are going to, quote, reclaim things, thank you, it's heavy, from the, let's put it this way. So here is the pyramid, and here's the medulla, and here is the cortex. If I'm telling you that we're going to reclaim things from this filtrate that we lost non-specifically, how are we going to do that, and where are we going to put this stuff that we reclaim? Well, we want to dump it back into the blood supply. So here is, let's orient ourselves. Here is a cortical nephron. Here's the Bowman's capsule specifically of a cortical nephron. Here we see the convoluted tubule system associated with that nephron. Here we see the collecting tubule system associated with that nephron. Note these capillaries draped over the collecting tubule system. They are reabsorbing or accepting the reclaimed solutes from those collecting tubules. Up in the cortex, that's called the peritubular capillary network. Peritubular in the cortex. Down in the medulla, we're going to give it a different name, vasa recta. Alrighty, so let's restate that. We have two kinds of nephrons, cortical and juxtamedullary. When we have a cortical nephron, the capillary net that serves it is going to be called the peritubular capillary net. And we have the equivalent in the medulla, it's going to be called the vasa recta. So something different. And you get the best description of that with this model in particular. Thank you. You don't see it in the same degree of detail with this model, although this is an excellent model in general. Yes?
What about the juxtamedullary apparatus?
That's what I'm getting to. There's a different juxta. Okay, so perfect segue. Let's now look at the Bowman's capsule. Somebody tell me from histology, you looked at these repeatedly, and they were on your practical in the histo portion of our quarter. We said that this kind of epithelium here lining the capsule is what kind? Simple squamous. What kind of epithelium is this? It's also simple squamous, right? Because capillaries are always simple squamous, and this is a capillary net. So we've got some funny looking cells that are wrapping around this simple squamous epithelium here. What are those? And they interdigitate like this, or dovetail. And you can see how they tried to represent that here on the model. Those are podocytes, foot podocytes, because they have these foot-like extensions. And they are reinforcing this simple squamous epithelium because we have filtration going on here and high pressure. If we have a high pressure system here, we're going to blow up those simple squamous capillaries, and then our kidney's not going to work. So we have to reinforce that given what goes on in this place. So blood comes in via our afferent vessel into this space, and the filtrate collects all around this glomerulus and filters out. What is this thing right here, this big green looking vessel? Well, as the filtrate is being created, it will pass by this afferent vessel, and they will physically touch each other. And there are special sensory cells here, mesangial cells and granular cells, that will sense the concentrate in the tubule system. If there is a lot of something there, then it, quote, tells the capsule, hey, maybe we're filtering too hard, because a whole lot of these things are being found here in the filtrate. So maybe we need to back off on this pressure so that we don't have so many of these solutes coming into the urine. And in that case, the smooth muscle cells right here in this afferent vessel will dilate or constrict accordingly. That communication between the afferent vessel and this part of the collecting tubule system is called, structurally, the juxtaglomerular apparatus. Okay? So you've got two juxtas. You've got a juxtamedullary nephron. That's one thing. And then you have this special apparatus called the juxtaglomerular, because it's near the glomerulus, juxtaglomerular apparatus, which is a sensory system that helps to fine-tune the filtration of the urine. All right, and it's completely different. Okay. Yes, I think if you have your list in front of you that I've hit on every item on your list. Is there something that I've missed that you have a question about?
The urethra and bladder are supposed to be seen on the kidney model, right?
Urethra?
Yeah.
They'll be on that model right there because you have your bladder, 6